We sort of left off finalizing the genetic code, talking about how the genetic code works. And we looked at what would come in, what base would, or what amino acid would come in for each codon. And so we're going through and looking at some codon sequences and noticing that AUG is always the start codon, but it also is going to still bring in methionine anywhere else that it occurs. So AUG will bring in MET wherever it happens. Okay. Does anybody have any questions about how the genetic code works? Because what I want to do next is move forward and talk about translation itself. So translation is going to use the genetic code, and it's going to use transfer RNA and ribosomes to bring all these pieces together with the messenger RNA to finally interpret the message into an amino acid chain. So in order for translation to get started, we have to have a mature mRNA, right? We have to have our messenger RNA ready to be translated. And then we also have to have our tRNA interpreter molecule ready to read that message. And in order for the tRNA to work, we're going to need the amino acids that charge the tRNAs. We're going to need the enzymes that are necessary to catalyze all of those reactions. And we're going to need energy because this is an energy consuming reaction. And then lastly, we need our ribosomes. Ribosomes are made up of ribosomal RNA and protein complexed together into 2 3-dimensional structures. And those ribosome pieces come together and like clamp together and hold everything in place and serve as the the workbench, the desk where this happens, like the place where we bring all this stuff together is at the ribosome. So you're going to start to see a lot of images like this one. In this image here, the messenger RNA is shown in pink. The amino acid chain that is being formed is shown in blue. Our tRNAs are here in green. And then the brown structures, and you can see that there's the light brown structure and the dark brown structure. Those are the 2 subunits of the ribosome. Okay, so everything is going to come together for translation to occur. The first thing that we want to say about ribosomes is that ribosomes are not specific. So we have the large ribosomal subunit and the small ribosomal subunit, and they are completely generic. Like all of the ribosomes do the same thing. They don't interact with only certain molecules. They just are generic and they work. Within the ribosome, we have 2 grooves where the 3-dimensional shape creates pockets. One of them is referred to as the A site, and one of them is referred to as the P site. The A site is where the new amino acid is going to come in. So A is the amino acid addition site. So the tRNA that slides into that A groove is going to be carrying a single amino acid, and it's bringing it to add it to the growing chain, the growing polypeptide. Okay. The P site holds the polypeptide polymer. So it holds the growing chain of polypeptides, the individual amino acids linked together. Okay, so the A site is going to have a tRNA that's carrying a single amino acid come into it. The P site is going to hold a growing polymer of many amino acids already linked together. Okay. So this is what this looks like. Down here on the bottom, we have the small ribosomal subunit. Then we have the large ribosomal subunit. In the A site, we have a single amino acid being brought in on a tRNA. In the P site, we have this growing polypeptide chain that we're going to add that new amino acid to. So translation, just like transcription, has 3 steps: the initiation step, the elongation step, and the termination step. Initiation is going to start in from the 5' cap. Elongation is going to read every codon and bring in a single amino acid for every codon, every 3 nucleotides. And then termination is going to occur when we get to the stop codon, which is before the poly A tail. All right. So initiation begins when the initiation complex forms. And all of these pieces are going to have to come together for initiation to happen. First, we have the small ribosomal subunit down here. And we have our messenger RNA, our mature messenger RNA. And then we have a tRNA. And this tRNA is going to come in and dock on the messenger RNA at the start sequence. Remember, the start codon is AUG. It's always AUG. AUG also codes for MET, right? So this amino acid attached to this tRNA is always going to be MET. Okay. Once this forms, the charged tRNA comes in, locates AUG, and then the large ribosomal subunit joins. And the two subunits come together kind of like a clamp. They hold the messenger RNA in place. The tRNA is slotted into the groove. And at this point, the tRNA is actually in the P site because it has the polypeptide that will be, right now it's only a single amino acid long, but it's carrying that chain, the beginning of that chain. Okay. So this is initiation. Elongation occurs as we begin to add more amino acids to this growing chain. So a charged tRNA is going to come into the A site. So here's our new tRNA. And the tRNA that comes in is going to be the one that is carrying the anticodon that matches this mRNA codon that is in the A site. So the tRNA is going to come in and enter the A site. And then the ribosome is going to carry out its enzyme-like reactions. It's going to function as a catalyst. It's going to catalyze two separate reactions. First, it's going to break the bond of the tRNA at the P site, freeing that growing polypeptide chain. And then it's going to form a peptide bond at the A site, attaching this chain to the new amino acid that comes in. So our tRNA comes in, we break this bond, and we move the chain over to the new amino acid that just came in. So our first amino acid that was brought in is out here. And each time we add a new amino acid, we're adding it to this end of the chain. Okay. And then once we've got the polypeptide in this position, the growing polypeptide has been moved over to the A site. Now the whole thing has to shift over one codon. So this tRNA is going to slide over. This tRNA is going to leave. I guess it leaves first, and then the next one slides over. And now we have an empty A site for the next tRNA to come in. Okay. And so obviously this is going to happen really quickly, right? The cellular machinery is moving lickety-splitly. So every time this bond is made, we just have a shift. Next one comes in, everything shifts over. Next tRNA comes in, everything shifts. And we just keep elongating until the stop codon enters the A site. Do you guys have any questions about that? So elongation is just this continual process of sliding the messenger RNA through the ribosome, reading it one codon at a time. When the stop codon enters the A site, then termination happens. Remember, we have three stop codons. It could be UAA, UAG, or UGA. When it enters, translation stops. And that triggers the whole complex to fall apart. So the large ribosomal subunit and small subunit dissociate. The polypeptide and the tRNA are both released from the P site. Everything just lets go. So we're going to break our polypeptide off. We're going to release the mRNA, we're going to release the ribosome. The ribosome is just going to kind of unclamp and fall away. And then we have this polypeptide free in the solution, ready to go be moved through the endoplasmic reticulum, moved through the Golgi apparatus, processed, shaped, cut, whatever it needs to become its final protein. Okay. At this point, when this polypeptide is dropped off of the tRNA, can you guess what kind of protein structure it has? Primary. Yep. Unless it begins folding while it's still in there, while it's still attached to the ribosome, which happens in some cases, it's going to come off in its primary structure. Then it's going to be folded and processed and turned into its final protein. Okay. This process needs to happen really fast in eukaryotes. We need to make as many products as possible. We don't want to just make one protein. We want to make hundreds to thousands of proteins, right? We need lots and lots of these enzymes or lots of these proteins to do work in the body. And so we actually usually see what is called a polysome. And a polysome is a series of many ribosomes translating the same message simultaneously. And so what we have here is running from the 5' end to the 3' end. This is our mRNA message. And you can see that we have 1, 2, 3, 4, 5, 6, 7 ribosomes represented in this image, right? As soon as there is enough room at the 5' end, the next ribosome comes in and clamps on and begins scrolling along that message, making a new polypeptide. You can see the polypeptide in red here, growing away from the ribosomes that are pictured and then released. When it gets to the end of the message and everything falls apart, the next ribosome is moving right behind it. So basically, I have like a chain of ribosomes moving down the message like cars on a train. Okay. This is actually a scanning electron micrograph of that happening, and you can kind of make out the message if you follow through the ribosomes. It's not as clean as it is, and I lost track of it there a little bit in our diagram, but you can see over here all of these proteins being formed, kind of trailing off of the ribosomes, right? So we're making lots of messages or lots of proteins for each message, and we're making them simultaneously, and we'll just keep cranking them out until we have the number that we need or until the message degrades and falls apart and is no longer useful. Okay.